Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. Today we are going to discuss the histology of the gallbladder and the pancreas. So gallbladder is the pear-shaped distendable sac with a volume of about 50 ml in humans, and it is attached to the posterior-inferior surface of the liver. It develops embryologically as a secondary derivative of the embryonic foregut and as an outpocketing from the common bile duct. So it is an organ for the synthesis, concentration and storage of the bile. So route of the bile is that it is synthesized in the hepatocytes and then it travels via bile canaliculi into the right and the left hepatic duct. Here you can see these are the hepatic duct. From here it enters into the cystic duct and stored temporarily into the gallbladder. Upon stimulation from the hormone synthesized from the small intestine, there is a contraction in the wall of the gallbladder, and as a result of this contraction, this bile is then expelled out of the gallbladder via cystic duct into the common bile duct and then enters into the small intestine at the major duodenal papilla. So no muscularis mucosae or no submucosa is present in case of the gallbladder. Large number of the collagen fibers and elastic fibers are present in between randomly oriented smooth muscle cells. It is either having the serosa or adventitia, depends upon the presence of the peritoneum. So in the fossa for the gallbladder, where it is located on posterior-inferior aspect of the liver, there is no overlying epithelium, there is no overlying peritoneum. So here the outer layer is the adventitia, and in other parts the outer layer is covered by the peritoneum where it is serosa. In addition to this, there are the diverticula of the epithelium which are seen present in the lamina propria, giving a false appearance of the glands in the lamina propria, and these diverticula have been given the special name, that is the Rokitansky ash of sinuses. In addition to this, sometimes the gallbladders are also having ducts of the Lashka, and these ducts are of the Lashka are actually the accessory hepatic bile ducts which are present and which connected to the right lobe, which connect the gallbladder to the right lobe of the liver. So next is the pancreas, and you all people know that pancreas is both endocrine as well as the exocrine function. In addition to pancreas, the liver is also both having exocrine and endocrine function, but the difference between two organs is that in liver both the exocrine and endocrine function resides in the same cell, that is the hepatocyte, while in case of the pancreas, exocrine portion is separate and endocrine portion is separate, that is islets of the Langerhans. So in this diagram you can see this is the section, the gross anatomy of the pancreas. The head of the pancreas lying in the C-shaped curve of the duodenum, and this is the body of the pancreas, and then the tail of the pancreas. You can also appreciate the main pancreatic duct which is present over here. Sometimes the accessory pancreatic duct is also present over here, which opens into the small intestine at the minor duodenal papilla, while main pancreatic duct opens in combination with the bile duct at the major duodenal papilla over here. So exocrine portion of the pancreas synthesizes and secretes enzymes via system of ducts that are essential for digestion and the absorption. In this diagram you can also appreciate the common pancreatic duct and as well as the accessory pancreatic duct. The function of the endocrine portion is to synthesize and secrete hormone into the bloodstream to regulate glucose, lipids and the protein metabolism. So exocrine portion of the liver is formed by compound acinar, a compound tubuloacinar glands. Each gland consists of the pyramidal shaped cells. These cells having a broad base and having the narrow apical surfaces. The nucleus is usually, you can see the section of the pancreatic acinar cell. Here you can see basally located nucleus in the pancreatic acinar cells, and in addition to this, large number of the rough endoplasmic reticulum and the free ribosomes are present in this basal area, which is giving the basal area strong basophilia, which is also called the gastroplasm. In addition to this raphendoplasmic reticulum, there is a supranuclear Golgi apparatus which is well developed in these pancreatic acinar cells for the packaging of the packaging of the material which is synthesized by this raphendoplasmic reticulum. In apical part of the cell, you can see large number of the zymogen granules are present, and as the zymogen granules stain with the eosine, so you can see the eosinophilia in the apical region. The periacinal connective tissue over here is minimal in this case, and is acini consists of 5 to 8 pyramidal shaped cells. The centroacinar cells line the lumen of the acinus. You can see another important and differentiating feature of the pancreatic exocrine cells which differentiate it from that of the parotid gland, which is also purely serous in nature. But in case of the pancreatic serous acini, the duct of the gland starts within the lumen of the acini and then it becomes the intercalated duct. So the cells which are present within the lumen of the acini are called the centroacinar cells. Differentiating feature of the centroacinar cells are that they line the lumen of the acinus, squamous shaped cells which lack ergastoplasm and apical granules both. They stain very lightly with eosine. This is a feature which help us to differentiate the centroacinar cells from the acinar cells in routine H&E stain sections. These centroacinar cells sometimes secrete large amount of the bicarbonate ion as well. So accessory digestive gland having a well developed system of the duct. First of all, there are the smaller intercalated ducts which are present. These intercalated ducts then empty into the intralobular duct, and these intralobular duct and then empty into the interlobular ducts which empty into the main pancreatic duct. So intercalated ducts are a smaller size ducts and they are lined either by the low cuboidal epithelium or the simple squamous epithelium. Then are the intralobular duct, and these intralobular ducts are lined with the simple cuboidal epithelium, size on order of the magnitude of the acini. So outer diameter of intercalated duct is smaller than that of the acinus, while that of the outer diameter of the intralobular duct is larger than that of the acini. Then the secretions drains into the interlobular duct, and these interlobular ducts are present in the septas. Large number of connective tissues surround these interlobular ducts, and these interlobular ducts are lined by the simple columnar epithelium. Another point you must remember over here that there are the no striated ducts in case of the pancreas, because we have studied previously with the salivary glands that the serous glands are having the striated ducts, but in case of the pancreas there are no striated ducts. So this is again your important MCQ as well. So another diagram in H&E stain section, you can see these all are the pyramidal shape acinar cells having basally located nucleus, and there is the basophilia around the nucleus, and in apical area you can see eosinophilia. This eosinophilia is because of the zymogen granules. In the center of acini you can see light staining cells which are the centroacinar cells. So you can differentiate between the centroacinar cell and acinar cell in simple H&E stain section as well. Another beautiful diagram stain with H&E section, in which you can see these are the pyramidal shaped cells having basally located nucleus and basal basophilia, also called the ergastoplasm, and apical granules which stain with the eosine, and in the center a light staining cell is present which is called the centroacinar cell. This is again the section of the intercalated duct which is lined by low cuboidal epithelial cells. Another H&E stain section of the exocrine portion of the pancreas. Here you can see large number of the secretory acini which are serous in nature, and here you can see the duct as well. This is the portion which is the endocrine portion called the islet of the Langerhans. Here you can see the blood vessels as well. Another H&E stain section in which this all the portion is the exocrine and this portion is endocrine, also called the islet. So this is another small islet which is present over here. This is the section of the duct which may be interlobular, present in the septa, and large number of the acini. These are serous secreting acini which are present in the exocrine portion. This one is the smaller intercalated duct, you can see over here. So this is the exocrine portion, another diagram stain with the H&E stain section, and this is the endocrine portion. And this exocrine portion you can see very well. The basally located nuclei are present, strong basophilia because of the basal basophilia because of the RER and the free ribosomes, and the apical granules which stain strongly with the eosine. Here you can see also the centroacinar cells which are present, and these areas show the islets of the Langerhans, which is the endocrine portion of the pancreas. Another diagram in which you can see the different ducts. This is the intercalated duct, then the intralobular duct, and then the interlobular duct. So exocrine portion, intercalated duct starts within the lumen of the acini, and it is lined by either low cupidal or simple squamous epithelial cells, and the cells which are present in the center of the acini, they are lightly staining, and these cells are called the centroacinar cells. So in this section you can compare the acinar cells from the centroacinar cell. In centroacinar cell there are no apical granule, their nucleus is usually elliptical, and the cell is a squamous shape. There is no basophilia as in case of the acinar cell. So now we are moving to the endocrine portion of the pancreas. This is the islet of the Langerhans. In endocrine portion, the polygonal cells which are arranged in the form of the cards, and in between the cards you can see large number of the fenestrated capillaries are present as well. So endocrine portion principal function is to secrete hormones that regulate the blood glucose levels. Islet of Langerhans are masses of richly vascularized endocrine cells scattered throughout the pancreas. You can see this is the islet of the Langerhans, separated from the surrounding exocrine portion by smaller amount of the reticular connective tissue. And three types of the cells, usually four types of cells even are present, the A, B, C and D type of the cell, also called alpha type, beta and delta type of the cells present in the islets. So this is the exocrine portion of the pancreas, in which you can see the polygonal cells, which are different types of the cells arranged in the form of the card, and in between you can see large number of the fenestrated capillaries are also present over here. So minor population of the cells which are forming the 5% of the total cells of the islet cells, and they secrete the pancreatic polypeptide, the vasoactive intestinal peptide, the secretine, motilin and substance P, etc. Another H&E stain section, in which you can see this is the islet of the Langerhans and the cards of the polygonal cells. Another important point you people must understand that different kinds of the, it is totally unpractical to identify different kind of the islet cells in H&E stain section. You cannot differentiate alpha, beta, delta cells in H&E stain section. To identify these three types or either four types of the cell, we have to stain with special stains used to stain these cells as well. So another H&E stain section, in which you can see the at higher magnification the islet of the Langerhans. Different stains with the Mallory Azan stain are like A cell stain red with Mallory Azan stain, the B cell stain brownish orange color with the Mallory Azan stain, and D cell stain a bluish color. Their cytoplasm show blue color. So in this slide there are no A cells present. Actually A cells are not taken in the section, so you can only appreciate the B cells which are present. These are orange shaped cells. The beta cells are B cells, and at periphery you can appreciate the delta cell. These are large cells present at the periphery, and these cells having the bluish color cytoplasm. So these are the special staining characteristics which help us to identify different types of the cells present in the pancreatic islets of Langerhans. So the stain is the Mallory Azan stain. Here I complete my today's class and also the histology of the gastrointestinal system. The references are the Basic Histology by Laikus and Siddiq, Histology Text and Atlas by John Quera, and Histology Text and Atlas by the H. Ross. So thank you very much dear students for your attention today. Allah Hafiz till the next class.